Hello, everybody, and welcome to our webinar, Exploring Millions of Clinico-Genomic Data Points from UK Biobank. We created this webinar to address some challenges that researchers face when starting off their plans to use the UK Biobank dataset in their research. If at any point today you have a question, please feel free to participate by typing your question in the Q&A box or chat on your Zoom dashboard. I am happy to announce your host for today is John Penn. John has more than a decade of experience applying informatics and computational tools to bear on biological problems, from in silico protein folding to supporting development of next generation sequencing clinical tests to managing production pipelines for the Regeneron Genetics Center. At DNAnexus, John helps scientists leverage the DNAnexus platform for secure and easy omics informatics in the cloud. Today, John will be giving a brief introduction to UK Biobank, cover some challenges that researchers face to navigate the data, and present solutions to maximize value from the data set to drive precision medicine forward. With that, John, why don't you take it away?
Fantastic. Well, welcome all, and we look forward to going through this with you today. So to begin, we are talking about the UK Biobank and how we are structuring the data from the UK Biobank in our Apollo solution. Let's talk a little bit about what the UK Biobank is. Which is, I think, kind of unarguably one of the most important genetic data sets that's been produced and will be around for the next decade at the very least. Not only because of the scope and breadth of it with the size of 500,000 participants and their deep phenotypic records and their whole exome sequencing, but because this data set is being made available to researchers kind of throughout the world through application to the UK Biobank itself. So back in 2010, an invitation went out to a number of individuals around the UK asking for their participation in this really grand research project that is the UK Biobank. For this, they designed very clearly a methodology to collect information beyond that which they would get from just an initial assessment, but by having folks even link their National Health Service records through the UK Health Service to this particular study. Now, even in their intake for initial assessments, they went so far as to define what the room should look like and how the spacing should be so that they could go through and collect each of these initial metrics for an initial kind of baseline visit for each of the participants, and all of the 500,000 individuals went through this. Here's a kind of a quick image of one of the stations where they're taking one of their measurements. They linked beyond just these initial assessments, the kind of three types of data from the UK National Health Service: the death registry, cancer registration, and the hospital episodic data. So all of this data is being made available to researchers who apply appropriately and request this data that they can then apply in their genetic and phenotypic research. Now, already in the data that's been released with just the genotype data and imputation data, along with the 50,000 exomes that were released for after being sequenced by the Regeneron Genetics Center and released by the UK Biobank, a number of publications have come out showing very clear utility of this data set in really advancing our understanding of a number of genetic diseases. So DNAnexus was first kind of brought into the UK Biobank data world with our participation in the delivery of the actual whole exome sequence data. So samples from the UK Biobank were sequenced at the Regeneron Genetics Center in Tarrytown, New York, where all of the secondary analysis, variant calling on the data was performed on the DNAnexus platform and then shared through our secure platform to the pharma partners who all helped fund this really impressive delivery of data. Now, we recognize, looking through this data, there's a number of challenges with 500,000 individuals. Looking at just their genotype data alone is daunting. When you then bring in their phenotypic data with over 3,500 dimensions to it in the UK Biobank, it becomes a whole other issue of scale in terms of being able to hold and then navigate through that data. And by navigation, I mean, how do you go through and design those cohorts that you want to build and run analyses on? How do you make those tweaks? How do you decide which of these phenotypes is actually important and going to be relevant and build you that cohort that you're looking for? And then once you've built this cohort and done an analysis, or you're doing an iterative analysis going back and forth between a biologist and a data scientist, where is this data living, and how is it secure? How can you make certain that you're handling things like HIPAA, GDPR? So we're talking about, again, health records and personal genetic data that's tied very specifically to it. So with all that in mind, DNAnexus has built our Apollo platform. Now, we built this on that secure kind of on-demand infrastructure that is the standard cloud infrastructure, and then layered in our own security and management for scaling for population genetic analysis. We know that this allows for clearer data management and that iterative data discovery process that really does lead to insight. Now, what we've created is, in our methodology, a manner to really get at the data kind of from two entry points in terms of your technological expertise. We have a beautiful user interface that allows folks without any command line experience to go in a drag and drop fashion and type ahead in search fashion, create their own cohort, slicing on any one of those 3,500 phenotypic dimensions that do exist in this UK Biobank data set. And because this underlying infrastructure sits on a Spark cluster, it's very efficient and very effective, allowing for the push button running of GWAS and other analyses, producing through our analysis browser even these beautiful Manhattan plots with variance across your cohort of significance with their p-value listed below. For those data scientists that really want to get deep into the weeds and are ready to kind of explore that data set beyond in a drag and drop fashion, we also provide our Jupyter notebooks. Now, our Jupyter notebooks sit atop a Spark cluster that you aren't required to manage. By selecting a cluster and choosing the number of cores you need, that's as far as you need to go in terms of configuring. And then you're provided with a very clean interface that allows you to launch either R, Python, or Bash kernels to operate directly on the UK Biobank data. So with that, let's jump into the platform and let me show you a little bit about how we actually do this live. So diving into our live demo, what we've landed on here is actually the landing page for the dashboard for the Apollo browser. What this shows is the name of the data set at which I'm looking, the number of individuals that I've sliced out of the total possible cohort with some options to download and save. Now, as you look down below, I have a window for dashboard, a samples table that lists all the samples in your current cohort, which I've kind of collapsed here is a little unwieldy at this point, and a variant browser that shows the allele frequency distribution in a lollipop distribution for the cohort at a genomic loci that you can specify either using genomic coordinates or an actual gene name. Now, for the dashboard, we allow you to create and move and manage tiles that allow you to slice cohorts across, again, any of these dimensions. Now, for this starting point, I have two tiles, sex and age at recruitment. But I can add tiles here, which will bring me to a window where I can either browse through the folder tree that describes every actual phenotypic dimension on which I can slice this data from its original assessment center up through online follow-up and their hospital inpatient data. Or I can search using, so I know I'm interested in ICD-10 codes, so I can search ICD-10. Now what this will do is go through all of the possible folder trees and pull out any notice of ICD-10 like a normal search function. But you'll notice some of these might not necessarily be what I'm looking for in terms of a tile to add. So say I select secondary ICD-10 codes here. When I double click on this, this opens a secondary window that'll allow me to evaluate the field and tile that I'm generating to make certain it's the correct one. So what I'm looking at here is secondary ICD-10. So this is actually not the main ICD-10 code. It's not the initial diagnosis. Looking at this, if I have any questions, I can even click out and link into the UK Biobank data record registry and evaluate this data field as it is. Or I can move back and select the main ICD-10, evaluate that to see if this is actually the tile I'd like. And as I know this is, I'm going to let this populate and then click Add as Tile. Now, I'm also, just in the interest of the healthy obese, going to add BMI as a tile as well. Again, I have two measures, body size measures and impedance measures. I can click on these and validate that they are the chart I'm interested in. Some of these actually also can be converted from a histogram to a box plot if it's appropriate in terms of slicing your cohort. For BMI, however, I think this is a much better distribution on which I can do that. So I'm going to add that as a tile. I'm going to close my window. Now, on my dashboard has my new tiles available, and I can arrange these as appropriate to make them increase their utility as necessary for me, and kind of a drag and drop functionality. So now I can go through and create a cohort. So say I want to look at men over the ages of about 55 with a BMI that's, you know, between or obese, at least obese to clinically or severely obese, who actually don't have metabolic disorders, right? So, or let's say who have asthma but no diet, but don't have metabolic disorders. So I can look for asthma, and I can see that actually in all of our fields, there are 776 individuals in our pseudo cohort who are falling under the categorization of asthma. Now, you can break those down further by their sub ICD-10 codes and select smaller cohorts. But even doing that, you'll see you actually lower your cohort number. But by doing this, I can actually shape a cohort even by my early selections. I know that if I select, you know, whatever Y40 to Y59 is drugs and medication, my cohort drops to 0. So here I'm actually going to just take chronic lower respiratory issues. And I'm going to add that as a filter. And now I have my filters populating across the top here. My cohort filters, my sex is male, my age of recruitment is between 55 and 75. Now I actually wanted my BMI to start at 30, so I'm going to click in and change that number. So that's now very specific. And my main ICD-10 includes chronic lower respiratory disease. But I did say I wanted to exclude metabolic disorders. So I can actually do another search through the tree. So I can exclude all metabolic disorders. So this will now shape a cohort where this exclusion criteria is enacted, and click save. Now, it's calculating my cohort count here in the top right. Now, because I've added that kind of two-part filter, it's likely going to shave my cohort down smaller than perhaps I'd like. So in a moment, I'll probably go back and eliminate the chronic respiratory diseases. But the calculating cohort will show me that of this cohort now, 77 of my 100,000 individuals actually fit the criteria of the dimensions I've described in this dashboard. Now, again, I can go back in and I can say, you know what, I just want to keep my and excludes, and actually change that to excludes metabolic. Search and that, save that. And now that I have a cohort that I'm happy with, if I validate that my cohort number, as it's about to be updated, is 6,349, much larger cohort. I might be happier with that. I'm happy with the distribution of the allele frequencies, and I'm happy with the variance that I'm looking at in the population allele frequencies across even known data sets. I can save this data set just by clicking the standard save button and call this demo cohort. Call these our cases as healthy obeses. They don't have metabolic disorders and they are above 30 BMI. So I'm going to save that. Now, what you'll notice is as soon as I click save, this start analysis button on the top right actually became clickable. Now, I've not defined a control cohort here and can run this against essentially the entire data set, or I can go back and modify even my filters. If, say, I wanted to, as reductio ad absurdum, is just compare males to females, I could remove the male filter and make it females. And I can actually change that and refresh my dashboard. And now I have 7,640 of my 100,000 individuals. But in my case control analysis here, that'd be a pretty flawed study. So I'm actually gonna not use that. But now what I can do is I've defined a cohort. So either saving this and by clicking analysis, I can start analysis, and this will drop me into an opportunity to run a number of tools. And what I'm actually going to launch here is I'll launch our Plato GWAS pipeline. And this will drop me into a number of a window that allows me the opportunity to set the parameters and inputs for this particular GWAS run. So I have my execution name. I'm executing it in my project here. I can specify if I like within my project where I'd like my outputs to land. I'll set that in results. Select that folder. I can now specify my analysis inputs. So I have now defined my test2 demo. Now this was that second cohort that I defined, and actually I don't want to use that as my case. So I can remove that, go back in, and now go looking through the cohorts that I've described or created, and at the top, I can find a cohort and select that data and populate my analysis with any previously created cohort. I can add a control cohort if I like, but it is not required. I can now give this a VCF files as input, so I can go into my data sets where I have my VCF files, and I will take my chromosomes 21 and 22, select my data. Now I do have a couple required fields here marked by these stars, so I can call this my one and my demo -1. And now that I've filled out both of these required fields, my Run as Analysis button is now clickable. I do have one last tab available, which is Stage Settings. This allows me to structure the instance types that are going to be used for the analysis. So I can optimize here if I know something I'm running is going to require more RAM or require more cores. I can adjust that here, or I can go with the default and then click my Run Analysis. Now, as we click Run Analysis, this will actually drop us directly into a monitor log for this particular analysis job. And this lives within a DNA Nexus project. Now, we can actually go to the main monitor tab to see all jobs that are running or evaluate just the job we've launched. Metadata around this job is captured and stored, but is available also through this dashboard, including the unique execution ID of this particular analysis, the provenance of when it was launched and by who, using what executable. And this executable is identifiable by a unique object identifier, and it is an immutable object. So to update this, even by name, will require a new object ID, ensuring provenance of any analysis. And I can point and link out directly to the folder in which my outputs are going to be delivered. So actually, as we were here, you see outputs are actually starting to be produced in my outputs folder. The GWAS data set is actually being described. It hasn't been uploaded yet to the project, but is associated with the unique job ID for a sub job of our Plato pipeline, including the output as it's going to be passed through our pipeline. So we can go through and view the log and see how that's working and evaluate by either standard error or standard out. We can also go through and view the information about this, which is metadata specific to the project and job unique IDs on which they're launched. We can view all inputs and outputs, including all of the parameters, including defaults that we haven't reset but were passed to the applet, and all outputs that have so far been generated. And finally, we can actually terminate. By clicking terminate, this will immediately shut down the existing or the current applet and any downstream applets that may be part of a workflow or pipeline. Now I'm actually going to do that currently. Terminate that. You'll see our job is being terminated. That will be reflected in the log and available again for provenance. Now, going back to our results, much the cooking show metaphor, I do want to show you an earlier analysis I had done around blood pressure. And so what this is bringing up the actual association browser that we've built around the results from a GWAS. So this was actually run on chromosomes 20, 21, and 22, evaluating kind of differences in blood pressure really between men and women. So it wasn't again, particularly rich analysis, but again, this is a spurious data set, so anything we would find wouldn't really matter. But what we have here is, again, our Manhattan plot across the top, and then as we scroll down, you'll notice the listing of variance IDs, which can be sorted by their p-value in terms of their associations. So with that, now to get kind of deeper into the weeds with our data, we have our JupyterLab notebooks. Now, I've actually launched one a little bit earlier because they can take a few minutes to spin up in the cloud. But these are very straightforward launch by going through our tools library and to our JupyterLab. You can actually move into the JupyterLab project and create a new JupyterLab notebook. Now, once those are up and running and available, they provide inside the monitoring tab in a project a clickable URL, open up with a Jupyter notebook that allows you to run either Python, Bash, or R kernels. So because this has been launched from within the DNA Nexus project, and nothing really exists in its home folder, it is, however, tied to the DNA Nexus project from which it's launched. So this allows us to then peruse and browse through data sets in even notebooks that have previously been created that are available within the DNA Nexus project structure, which is kind of our overall security boundary. So moving into some notebooks that I've actually created prior. So what I'm going to show you here with the notebook is going through how we can build a complex cohort utilizing the UK Biobank data set, but going from a command line really kind of deep in the weeds angle. So actually in this first box here, we have a wheel file. It's a Python utility file that we provide that will allow you to kind of operate on the data as it's structured on our underlying infrastructure. But you'll notice I import this DX data, which is a kind of helper library for Python, but then standard libraries such as pandas. Now, walking through this notebook, if you've not used Jupyter Notebooks before, they're broken down into blocks where I can actually add markdown blocks of either code, markdown, which is just text, or even just raw text. So in a markdown box, I can provide information around how to utilize a command block below, or comment out in this kind of standard scientific lab notebook methodology. Moving through into our blocks of code, these can be executed either in single or actually kind of in serial by launching from the run command at the top. Now, as we walk through this particular notebook, what it does is actually load some standard Python packages along with our DX data package, and allowing you to kind of get into the meat of the UK Biobank data set. So moving through loading that data set into the instance on which we're working, because this is the actual compute instance running in the cloud, the Spark infrastructure underneath. We can extract traits from that, doing standard SQL queries, find out the structure of this kind of data, and select those pieces out of a huge return from a kind of almost database dump of all the possible fields. We can even then take out of those traits, those traits that we find are relevant. Define, again in Python, our set of conditions, and convert this score, creating our CHADS S2 score that we could actually run and evaluate across this entire data set, and upload back into our project. Further, even just querying results or doing GWAS results can be done either in R or Python. Again, our launcher allows you to launch notebooks in any of these languages. And even when working in a given notebook, you can actually create blocks of code in these notebooks that are different languages. So you can run Bash in the Python, you can run Python in the Bash with a little bit of, with just a couple of precursor characters, letting the application know that there is a change in kernel. So with this, I want to very quickly show you. So here's a exploration of GWAS results with some annotation going on, but this is all done in R, providing a QQ plot, again, our Manhattan plot, allowing for, again, really deep integration and interrogation of the data, using a tool set that many statistical geneticists are very, very comfortable with. But then our data scientists who prefer perhaps to work in a bit of Python have the same opportunity to get very, very deep into that data set with libraries that they are comfortable and familiar with that allow them to generate the results and annotations that allow for iterative discovery between our data scientists and our downstream biologists. So with that iterative discovery, going back and forth between data scientists and biologists, as you identified genomic regions that may be of interest or even variants, you can even go back and add a genomic filter to your analysis. Filtering by SNP, indels, mixed, you can actually list up to 5 gene regions, either through the gene name or their actual locus. You can select all high impact as we've defined them, or remove subsets as appropriate, and apply that, or apply by filter by variant IDs and apply, and that will then actually also reduce the dimension, or sorry, to further refine the development of your cohorts for your next analysis. Again, feeding back into the Jupyter Notebook, which allows you again to securely look at that data once again through our Apollo browser. With all the tools Apollo allows researchers to bring to bear on the analysis of UK Biobank data, we're really enabling iterative discovery of human genetics that really are important and empowering for the discovery of new biomarkers, validation of drug targets, and really for just understanding the links between genotypes and phenotypes. If you are interested in piloting our UK Biobank solution on our Apollo platform, please reach out either directly to me or info@DNAnexus.com, and you can find more information on our website. All right, so now that having been said, let's why don't we open it up for questions? I know there were a couple that came in during the talk.
Thank you, John. So the first question is about the requirements to access UK Biobank data.
So access to UK Biobank data is actually controlled specifically by the UK Biobank. Data access is acquired by applying specifically with them, and applications are granted kind of through their mechanisms.
All right, great. Can you talk a little bit about the software used to call variants, and what is the depth of the whole exome sequencing?
So there are 2 different pipelines that were used to call variants across the UK Biobank data. One was the Genomics PLC WeCall and DNAnexus joint genotype pipeline that was done at the RGC. The other was a functional equivalent pipeline that used the functional equivalence alignment from the paper that came out about 2 years ago now, and did variant calling through GATK, but still had joint genotyping through GeoNexus. The coverage overall, I know the average coverage was targeted to be at least, I think, 25x, I think it's 30x minimum across really like 80% of the exome. RGC is very, very effective at sequencing very well.
Wonderful, thank you. A question about, is it possible to load up other data to compare to UK Biobank Cohort, for example, a replication study?
So data can be uploaded to the platform. It's not as straightforward necessarily as bringing it directly into your Apollo. Is the data structured on a kind of Spark infrastructure underneath. And for effective and efficient querying of that data, we do currently suggest you engage our business services group to help ingest that data and make it really accessible for you in a really facile way.
And John, next question is, how flexible is batch analysis? Can you give a few examples implemented in frequently used workflows?
So batch analysis, so DNA Nexus platform really works kind of like a crank. And our whole idea has always been to abstract away from users the need to manage and secure compute resources on the cloud. And to that end, what we do is once you've codified a workflow or analysis, really it's just it's a crank. And as many times as you turn that crank is how many times those analyses launch off. I mean, you could launch in parallel as many analyses as you're interested in running, with really the breadth of the cloud just being the only potential limiting factor. And that's near, as far as we're concerned, nearly infinite, essentially.
Great, thank you. Are the GUI commands backed up by equivalent code generation? For example, if you create a filter, does it generate code you can manipulate?
Not currently. The underlying code, the find a cohort is not generated as a secondary file. You can, however, store data or create that in a notebook and recreate that and see very clearly, kind of in a human, as human readable as code can be fashion.
Great, thanks. A couple more questions here. Can you add external libraries with ease to the notebook, or is it more a closed off environment?
No, you can add any libraries you like. Very early on, we'll warn you, you're kind of responsible for any repositories or things that you add or libraries. We don't obviously keep up to date on, we don't keep Python libraries up to date, but yeah, you can upload anything, any tool you need. Our platform's designed to enable you to do things, not to hamper your ability.
Wonderful. Can we access Genomic Viewer for the pileups of the BAM data files?
So our platform provides a number of viewers through the user interface. The web versions like UCSC Genome Browser and with their JavaScript browser and IGV, and look at BAM read stacks through IGV on the platform. And that is available through kind of any project on the DNAnexus platform.
All right, perfect. And last question today is, do you have workflows for PheWAS analysis?
There aren't currently any canned PheWAS analyses that we have available, but any PheWAS pipeline that you want to bring to bear, it is possible to wrap and bring to the pipeline or run from a Jupyter notebook in a very, very clean fashion.
Great. Well, thank you so much for going through an in-depth tour of the Apollo for UK Biobank browser. If anyone has any questions, feel free to reach out to John. Thank you. Hope you have a great day.